A recent paper claims to have reconciled the fossil record of sponges with molecular clock analyses by finding that spicules were all biomineralized independently in each class, and sponges originated in the early Ediacaran. Join us as we explain why we don't think everything is quite as it seems. Previous videos have covered some of the problems in interpreting the sponge fossil record and the reasons why some of us are seeing homology of spicule skeletons across at least 2 and maybe all of the living sponge classes.
This is a critical question because if you look at the molecular clocks, which estimate the origination times of organisms based on mutation rates of genes, which you calibrate using fossils, the fossil record, and those molecular clocks predict a very deep history of Precambrian sponges that we are not seeing. So it'd be something like 100 million years of missing sponge record. You can explain that if all the early sponges didn't have a skeleton, they didn't have spicules, they didn't have biominerals, and they didn't become fossilized. But if some of the classes share spicules, say demosponges and hexactinellids, then you should have a deep history of spicules, and then that would mean that the clocks were wrong. And there must have been very rapid evolution in the lead up to the Cambrian Explosion. So there would be something going on that we don't understand. So it is an important question.
It is. It's not just about the sponges, although that's important anyway.
Oh yes.
Yeah. So enter the new paper. At least 4 or 5 people have actually drawn my attention to this, but it turns out we both knew about it already, because we have some history with this one. In fact, we've each reviewed it for different journals, and we've each raised major objections to it when we've seen it. The first time round it was rejected, the second time round it's got through anyway.
And just to say that the review process is normally anonymous, but we as reviewers can choose to say, yes, we reviewed the paper. We can't necessarily provide any details of what we said during that review because that would be confidential, but we can certainly critique things that are still there in the published paper.
Yeah.
That's fair game.
Yeah, so it makes sense to do it this way, and it just means that we've actually spent quite a lot of time thinking about this paper, so we know it reasonably well. So, it's been published in Science Advances, and this is a major open access journal from the Science stable. Open access is always nice, of course, but it does mean that the author pays an article processing charge, which in this case is $5,450.
Which is nice if you have that sort of money, and a big research group does, of course.
Yeah.
And it does mean that everyone can read it.
Yeah, and there are exemptions for people in developing countries and so on, but for most people like us, it's just not a place that we could publish. But this is quite a big research group, they have the funds, small change for them, so here it is. It is by Rossi et al. It's based on, we understand, a PhD, and there's a wider research group that's been working around this as well. So this paper provides an extensive phylogenomic study. So this is looking at a molecular analysis of the entire genomes of many sponges, and other things as well, and calibrates the tree using fossils to generate a molecular clock. They then look at 2 calibration strategies, so one in which spicules are shared across all sponges, it's their SPN model, and another where the demosponge spicules originated within the Cran group Demospongii, the SDN model. This kind of becomes important later. And they're providing results in this tree from the latter. So this is the results of the latter situation where they're assuming demosponge spicules originated in Demospongii.
So the common ancestor of hexactinellids and demosponges didn't have spicules.
Yeah, exactly. So in this scenario you have an early Ediacaran origin of sponges and an early Cambrian origin of spicules, which is more or less in accordance with the fossil record.
If spicules originated independently in all the sponge classes.
Yeah. And so far this is fairly standard, that you take a large genetic data set, you calibrate it with fossils, and you get a molecular clock, and it gives you some answers and you interpret those answers as you see fit.
Yeah. But then there is a logical thing you can do if you're considering the fossil record, which is to look at fossil sponges. Because if you're worried about what things look like ancestrally, why don't we look at some of the ancestors that actually got preserved, i.e. the fossils, which we have quite a few of.
And this is also what they do in this paper.
Yes.
So
Good.
they've extended this to an ancestral state reconstruction analysis, where they add in a handful of early sponge fossils, incorporate this into their phylogenomic analysis, and use this to reconstruct what they think the ancestral sponges in various groups, various parts of the tree, actually had as a skeleton, whether they could secrete silica, calcite, and so on. And it all sounds pretty good.
Sounds good.
Yeah.
Yes.
It's actually a very impressive looking paper. It's a very technical approach. There's a huge amount of backup data online in a Figshare file, which we ended up downloading about 1 gigabyte in order to try to piece together what actually happened in this paper.
And we should say we don't necessarily have problems with the methods. First, because we've got no reason to think they're applying them badly, and second, because we're not qualified to judge those methods.
Certainly not the phylogenomic stuff.
No. What we are qualified to comment on is fossil sponges. Very qualified indeed.
Yeah. So in this case, what we've got is a paper which is using state-of-the-art techniques and is generating a large phylogenomic data set, is using previously existing data on some fossils, which we'll come back to, coding them in a new way as well in some cases. And it turns out that it looks like this is a really shiny new paper which does what it says on the tin and gives you the answer.
Which would be nice.
Unfortunately, we think it's a bunch of baloney.
Yes.
It's a combination of, how should we put this, garbage in, garbage out, and if you plant tomatoes, you're likely to get tomatoes.
Do you mean tomatoes?
Oh no. Well, maybe.
Did you just make that one up?
Yes, absolutely.
Well done.
Lots of these problems are because so much of the important background, the assumptions, the exact protocols and so on are hidden in data matrices, and they're in supporting information, they're in Figshare files that you have to follow one link to get to another link to get to the actual data. It's not easy to find the actual information that generates these results. And once you follow all these and you spend quite a while peering through it, you start to realise that there are a lot of problems, some of them fundamental and absolutely catastrophic, and others much more minor, but when you've got a lot of minor ones, they build up. So we're not going to go through everything, obviously, but we do want to give you a flavour of it. So we've actually got notes for this one, because there's just too much to try to keep in our heads at one go. Right, so what have we got to start with?
Well, there's a calibration point they're using for the earliest demosponge spicules, which they're using as latest Ediacaran, saying silicious spicules. And there's a couple of problems with this. The first is it can be really difficult to tell the original mineralogy of spicules, because silicious things can get preserved as carbonate, carbonaceous things can get preserved as silica, anything can get preserved as pyrite, anything can get preserved as mould, and there's been a lot of assumption in the past, quite reasonable assumption at the time, that if you see a hexactine, for example, it must be silicious because all the modern ones are. And we'll be mentioning this again later, we know better than that now, that it doesn't have to have been originally silicious. So that's one problem. Assuming that these early spicules were silicious is very much an assumption, but they're stating it as a fact. That is one problem. They got some citations, but they don't actually demonstrate what the original mineralogy of those spicules was. And also, the spicules they're citing as earliest demosponge spicules, the reference they're citing for that is a paper that doesn't actually mention the word spicule. I've checked the whole paper. It mentions demosponges, but it doesn't mention demosponge spicules or their age. And I think it is fair to say that I pointed out that this was a wrong citation to the authors well before publication. So I don't know why they are citing that paper, because this is a very important point that their analysis actually depends on, so it is crucial that they justify it.
Yeah, it's one of these. I think because the difference in time between that and definite demosponges is very small, it doesn't make that much of a difference practically, but it's the principle of the thing.
It also, I may be being very unfair here, but if someone's making an obvious mistake like that, you wonder what else they might be being sloppy about.
Yeah. So, and this whole sort of review we're doing is not going to be small details like that in most cases, but there's a lot of details like that as well.
Yes.
So that's slightly worrying. Okay, let's go on to the phylogenomic part. This is not our area. It looks pretty good, although it's completely dominated by demosponges. As in, they have dozens of them. Next biggest group, the hexachinellids, represented by, I don't know, they've got 3 hexachinellids, 4 calcareans, and 2 homoscleromorphs. So it's very, very biased towards demosponges.
Right, just a quick reminder for the sponge fans.
Mm.
The 4 extant living classes of sponge are the demosponges, which are the most numerically abundant, the hexachinellids, which are the most lovely, the calcareans, which are calcareous, and there's the homoscleromorphs,
Which are weird.
Yeah, that's a fair point.
Yeah, so those are the 4 living classes, and demosponges are the most diverse.
And both demosponges and hexachinellids have got siliceous spicules, so the homoscleromorphs, no one's quite sure how they evolved because they have virtually no fossil record, and the calcareans have got calcareous spicules.
Yeah, but when you're looking at a molecular clock for demosponges, that's fine to just really focus on that. But the problem is that the hexachinellids, for example, they have 3 species included, and they're all in one derived family. It's telling you about that family, not about hexachinellida as a whole. It's just an odd choice. So it's limiting the data availability.
I mean, the data available for hexachinellids is limited.
It's not that limited.
Okay, fair enough.
Yeah, it's
Yes.
it's an odd choice, but on the other hand, it's probably just about workable when you're looking at the deeper nodes, so the distribution of features between classes. So it's okay. It's just you'd expect to see more of an even balance for this sort of work, maybe. But anyway,
Yeah.
So they then calibrate these this phylogenomic analysis with some fossils to give you a molecular clock.
And this is the standard way of doing it.
Yeah.
That. The method's fine.
Yeah. And they have 8 data points for the sponges, which is not very many. And again, they're slightly odd choices.
Mm.
So there's quite a lot of relatively young ones. Most of their calibration points they've chosen are Mesozoic. So subgroups of demosponges, for example. And which, you know, it's fine. It's again a little bit limited. And you would think that this might tend to avoid giving much deeper roots, which other molecular clocks have done, potentially. It's slightly odd. And then they're conflating the crown group hexachinellida. So they have a calibration for crown group hexachinellida, which is a Rossellid, which is fine, but then why wouldn't you use that as a calibration point for the Rossellidae rather than for hexachinellida?
Given that there are lots of Paleozoic hexachinellids, that everyone agrees are hexachinellids.
Yeah, some, yeah. But it's just these ones, you know, are actually Rossellids.
Yes.
So it's again, I would want to sit down and talk to them about the calibration points, I think, because some of those are a little bit odd. But this is all not the main problem with the paper. These are all just sort of little niggles that we'd want to see ironed out, really. So they then add in some fossil taxa into a combined phylogenomic dataset with cladistic dataset. So this is not looking at the genetics, of course, because you can't for fossils.
Yep.
It is taking a dataset based on a morphology matrix. So you list characters of sponges against sponges, and you create a data matrix of which characters occur in which one. And then this part of the phylogenetic tree is then incorporated into their larger phylogenomic one. So they're different analytical approaches, but you can sort of slot one into the other. Okay, this is where it starts getting interesting. So the data matrix that they have for the fossils is, and this is the one that they used for the ancestral state reconstruction. So this is the critical part of the paper. The paper is about reconciling the fossil record with the molecular clocks.
Yep.
So you have to really focus on the fossil record part if you're going to claim anything like reconciling it. So the data matrix that they have for this is not one they've put together themselves. They've taken it directly from a paper by Wang et al., which was put in after review, I believe, for Helicolochellus. This is this supposed hexacnellid sponge in the Ediacaran, which we haven't got onto yet, yes, we know.
It's going to be a separate video at some point.
It will, yeah.
Yes.
But we have concerns about it, which included a very small and highly deliberate selection of fossils, so I think it was 7 species, that were inserted into that paper basically to show the resemblance between this thing and some fossil hexacnellids.
Yeah, so that was an analysis for a particular purpose for that paper. And it's absolutely fine that Rossi et al. Are using that data matrix as the basis for another analysis. People reuse data sets all the time, that's fine, that's why data sets are made available. What Rossi et al. Have chosen not to do is they've chosen not to put in extra fossil taxa. So you'd think that if you were interested in the ancestral state of fossil sponges, whether they had minerals or not, whether they had spicules or not, what minerals they had, you'd want to include as much information about fossil sponges where you know the mineralogy as possible. So you'd think that the right include, say, Cardui-spongia, which we know the morphology, we know the mineralogy, and we even know the soft tissue.
Mm-hmm.
They might want to include Linica, where we know it had both calcite and silica in its spicules, in the same spicules. They've chosen not to include those 2 taxa.
Whereas what they have included amongst these 7 taxa are 2 genera which are synonyms, almost certainly.
Mm-hmm.
One is probably a fragment of the other, and this has been published, not in detail, but it's in the literature as they're probably synonyms. And then 2 taxa which are where the mineralogy is not known at all. One you can very safely assume it, but we'll come back to that in a bit. And it's just a very small selection of sponges which are very disparate.
Yes.
They are very different in their morphology. So, yeah, before we come on to that, I just want to say that what we don't know is whether they have changed the coding in this matrix. Because I eventually found the data matrix. It was in this near gigabyte of files it downloaded. And it's a Nexus file, which is one that you need certain types of phylogenetic analysis software to be able to open, which I can't do.
And this is a problem. I think that the ideal, the best practice is to provide your data files as a comma separated value file, which can be opened in all sorts of software. So that in 20, 30, 40 years' time, when the phylogenetic software that everyone uses is different and might not use Nexus files, people can still read it. And people who don't have phylogenetic software can read it right now.
Yeah.
So we haven't been able to check their coding, and that in itself is a problem.
Yeah, because
we should be able to.
We know in the previous version of this by Wang et al. There were some problems with the coding.
It's fair to say, we can talk about the peer review for that because it was open, that they did correct the data matrix between iterations.
Okay, good. Right, that's something,
yes.
Yeah. Okay. Right, so what we're getting onto, though, is the crux of it now. Because we now have these 7 species, which are highly divergent. They are totally different forms in some cases. But there's also a lot of uncertainty in what the morphology and mineralogy of these things was. So what you end up with, if you have just a handful of very divergent fossils included, and you're doing a phylogenetic analysis of these based on a cladistic matrix, is that what you're lacking is all of the continuity. You don't have the things that link different sponges together. You just have oddballs off on their own. And so 1 or 2 characters completely skew the analysis. And you can see that this has gone wrong.
Well, for example, they included Helicolochellus, and okay, it's been claimed to be a hexacnellid. We disagree. We'll get into that another time. But Helicology is interpreted as having non-mineralized framework. I can't even remember whether they interpret it as having spicules or not. It all got a bit confused. But so you've got a hexactinellid that isn't mineralized in your analysis. Now, if it isn't a hexactinellid, you've just biased your analysis by including something that isn't a hexactinellid. This is a problem, potentially.
Yeah. So only what you can do is run sensitivity tests, and they did this for something. So they remove some bits of data, try it again, see what difference it makes. They didn't try it for this, which is one critical thing.
Yeah.
But we know that this analysis has gone wrong because of a couple of things. Firstly, the position of Voxia. And Voxia is a Burgess shale genus, which is very well studied. It also occurs, or at least versions of it, related and similar genera in China as well. And we understand its morphology quite well. We understand its mineralogy. And it's even its biochemistry to some extent, in that we've shown that it has a chitinous fibre skeleton.
And there's an awful lot of disagreement about the interpretation of various early sponges. And Voxia is quite unusual in that everyone who's working on early sponges agrees it's a demosponge.
Yeah, and it's very clearly allied to the Virungida. So it may be slightly more basal, it may be leading to the Virungida, and possibly the split with the sister group as well, the keratosans.
For the people who aren't yet up to speed with sponges, Joe, what are the Virungida? Just remind us.
Yeah, it's one of the major. It's one of the major groups of demosponges. So you have the division into demosponges with spicules and demosponges without spicules, and the ones without are the keratosans and Virungids. Virungiomorphids, anyway. Yeah. So there is no ambiguity over Voxia. We know what it is. Some of the Chinese specimens also show silicification of these fibres, which is a bit odd in some cases, except that the Virungida also show silicification of the fibres in modern ones. This is all published. However, that wasn't included in their data matrix for the modern sponges. They said it was non-biomineralized, which of course leads to a bit of a skew anyway. So as a result of all this, Voxia turns out as a stem group Cilician, so a sister group to hexactinellids plus demosponges.
Yeah. So if you've got somewhere where you already know where it should go, based on your pretty good grounds, and it's not turning up there, you need to be a bit suspicious of the rest of your results as well.
Especially when one of the other things you got is Iphelia, which is a heteractinid. It's a very interesting heteractinid because it also has hexactins. Heteractinids are a group that are pretty universally, although not on totally watertight grounds maybe, agreed to be stem group calcareans.
But Iphelia was the only heteractinid-like sponge included in their data matrix, so there is no way that Iphelia could have come out with Calcarea, even if that's where it should have gone.
Because you don't have the morphological continuity of other taxa. So that again ended up in a spurious place. I think it was as an early hexactinellid within the crown group of Cilicia.
Bottom there somewhere, we're not quite sure.
Yeah, and it's because of the lack of data in this matrix. It's the lack of taxa that actually link everything together.
Yeah. So again, we know there's more information there. We know that you've got these heteractinids that have almost certainly original Calcarea spicules. Some of those spicules are like some of those of Iphelia. There's this link to Calcareans, and that can't come out of your analysis because you're not putting in data that we know exists.
Yeah, absolutely. And there's no reason for Voxia to come out as stem Cilician, apart from a really bad analysis. Okay. They do actually address why there are so few taxa included. So I'll quote from the paper. Ideally, they would have wanted more, dozens more. But what they say is, unfortunately, the majority of fossil sponges have never been included in a formal phylogenetic analysis, and their relationships are unknown. Accordingly, they cannot be included in ancestral state reconstruction analyses. This is very interesting sort of assertion. On the one hand, it seems to be saying that you can only know a fossil's relationships if it's been through a formal phylogenetic analysis. And nobody's done that with Vauxia, so I guess we can't know it's a demosponge. Yeah.
But maybe it's not a demosponge. We can see the answer there.
That is true. That is true, yes.
Yeah.
So, on the one hand, it's saying that a lot of the work done by generations of paleontologists is pretty much worthless. And then following that with, so nobody's done this work of coding the fossils yet, and therefore we can't possibly do it either.
And they're using the data matrix of Wang et al. As a basis. They've got a list of characters. They could have chosen to code up some extra sponges using the same framework, and they chose not to do that. And they might well have had a very good reason for not doing that, but we're quite entitled to criticise them for not doing it.
Because that is, if you were doing this as a bit of an aside on top of a paper based mainly on the phylogenomics, no problem. But this is a paper that aims to reconcile the fossil record with the molecular clocks, in which case you need to actually include the fossil record.
And ideally as much of it as possible, not just the convenient bits.
Okay, let's move on now.
Yeah, let's
yes. So, going on from that, the ancestral state reconstruction is a different type of analysis which has been superimposed onto this phylogenetic analysis. So here what they've done is put the characterise the end members of their bits of the tree, including the fossils, with the mineralogy that is known for these fossils, sponges, living ones too, and then have an analysis which estimates the history of those based on the ends of the branches and what is shared and what isn't shared, and traces that back through time to give you an answer for whether the biomineralization was independent or not. So it's another, I think it's a fairly basic type of analytical method.
Yeah,
based on what we can see.
Again, we don't have a problem with the method, it's the data.
Yeah, I think the method's got problems as well for this. But
well, maybe, but yeah.
Yeah.
So,
right. So, one of the problems that I can see with the method is that, for example, it seems to be including a heavy bias towards being aspiculate and not having spicules. And you can see this when you look at their hexacnellid part of the tree.
Which, let's remind us, the extant taxa are 3 rossellids, and then they've got some fossils as well.
Yes.
Yes.
3 rossellids. So this is within one family in the crown group of hexacnellids, every single one of which secretes silica spicules in a coherent arrangement, and you end up with, well, we know that silica spicules are shared across hexacnellida. None of them have ever lost them as far as we can know. Yeah. So it's slightly surprising when you see there's a significant chance, according to this analysis, that the last common ancestor of Rossellidae didn't have spicules. And in fact, members of the crown group of Rossellidae didn't have spicules.
That's a ludicrous answer. That's got to be wrong.
Yeah.
Because Rossellidae is just one family of extant hexactinellids. You really need to start worrying about how good your other answers are at that point.
Basically, what I'm worried about is what systematic bias there is in here. Because it seems to be smearing out the probabilities from some sort of basal point where you say, if you have some point in the past where there was no spicule, it smears that out towards the crown group where you know there were spicules, but it's still saying there's a chance there were no spicules. And how that can influence the analysis of the tree as a whole is worrying, I think it's fair to say. So bearing that in mind, that there are these potential issues with how the analysis runs, and exactly what the method does, I have no idea. I haven't looked into the code. I'm not even a coder.
Yeah.
But yeah, it's that sort of level of worry that I have with this type of analysis, where there is a lot hidden in the algorithms because I don't understand what it is being done. Okay, so yeah, so where the mineralogy was unknown, that's in 2 of the fossils, it seems to be encoded, as far as we can tell, as 25% chance of each of the 4 options of no mineralogy, calcite, silica and bimineralic.
So with both calcite and silica.
However, that's not how they say they've coded it in the paper, where bimineralic isn't a separate option. It's calcite present or absent, silica present or absent.
So we can't quite work out what they've done here. And it does look like they chose to code the taxa that they didn't know the mineralogy of equal chance of any of the options rather than unknown. So if you're introducing the possibility that you could have calcite or you could have aspiculate reticulars and relatives of hexactinellids, I think there's a problem there, isn't there? Why not just leave those taxa out if you don't know?
Especially when you're doing an ancestral state reconstruction.
Yes.
And it's particularly amusing when you look at Diaganiella and Protospongia, which are almost certainly synonyms in that there is no complete Protospongia specimen, and it's probably a bit of Diaganiella that's gone like that. So they are almost certainly synonyms. And we know, well, this is another awkward one, because there is an abstract published that claims that Protospongia is calcite spicules, rather than silica, which everyone had assumed until that point. We've questioned whether it might possibly be bimineralic on the basis of there being axial structure that's distinct in some published images, but we are still waiting for the actual paper to be written and published, which shows us convincingly what this mineralogy was.
Yeah, so Protospongia might be calcite, might be bimineralic. It's possible that some of it might have been silicious. We don't know. No one's done the work.
And it's coded as being bimineralic.
And the article that they cite for Protospongia being bimineralic is the original description of Protospongia by Salter in 1864.
Yeah.
He does not discuss the mineralogy.
Don't
He's certainly not saying it's got calcite and silica. So this is, again, they're being sloppy. We don't know where they're getting that information from. They're not justifying it properly.
Yeah, so that's one issue. What I was going to was you've got these two species, two genera next to each other, Diaganiella and Protospongia. They've coded Protospongia as bimineralic. They've coded Diaganiella as unknown, i.e. 25% chance of all of them. And the analysis has come up with the last common ancestor of these two genera, which remember are probably one genus, as having a 50% chance of being bimineralic, about a 30% chance of being silica, and a small chance of either having no skeleton at all or being calcite. It's just, I'm not convinced by this analysis actually doing anything useful at this point.
Yeah. And again, they've chosen not to include some very relevant information, which is Cardiospongia, which is a reticulosum, like Protospongia, like Stylophycus. We know it was originally calcitic.
Yeah, and we have things like soft tissue structure, which is related to calcareans as well.
Yeah.
So there's just so much that wasn't in here, but we kind of put that to one side for the moment.
Yes.
We're just looking at the actual ancestral state reconstruction. So there's one other really key aspect here, which is that they have two sets of analyses for this ancestral state reconstruction. For one of them, they allow different metabolic pathways leading to mineralization in modern forms to be homologous. And in the other, they say that different pathways must be non-homologous spicules. As
far as we can work out, because we can't see the raw data, but we think that's what they've done.
Yeah, pretty certain. I think it's fairly clear. They've got, and they do spell it out. So they've got these two types of analysis. And this is based, I think, largely on the work of Shimizu et al. A couple of years back, who looked at the biochemistry underpinning the silicification of spicules in demosponges and hexactinellids, and found that there were different enzymes that were involved in that secretion process.
And this was a really important and useful piece of work. It told us a lot of things we didn't know already. What we have to be careful of is looking at what goes on at the present day and assuming we can just put that back into the past and it was the same.
Yeah, the conclusion of that paper was, and therefore they mineralized their spicules independently. But if you take that assumption at face value, then It means that various types of mollusks, for example, within one family, evolved their shells independently.
It turns out that mollusks use a surprising diversity of shell secretion enzymes and things, even in quite closely related taxa. So you have to be a bit wary of the assumption that sponges could never have done anything different.
Basically, the more complex our knowledge gets of the secretion mechanisms of biomineral skeletons, the more we realise that there's normally a suite of chemicals involved, and the exact balance of the enzymes involved in one group versus another can shift completely. So you can have completely independent suites of enzymes which are secreting the same evolutionary structure. So you can have a shell which has continuity through long evolutionary history, but has different enzymes involved in secreting it. And there is no reason to think that that wasn't the case in sponges. In fact, the Shimizu et al. Paper showed that in some groups of hexacnelids, the outer part of the spicule is secreted by a different enzyme to the outer part of different groups of hexacnelids, which demonstrates that there has been flexibility. And of course, this also completely ignores large chunks of the fossil record, like Conciliospongia cythoficus, where you have a hexacnelid-like skeleton with demosponge diagnostic axial canals. And those canals are based on the symmetry of the packing structure of the enzymes.
So it does tell you something about what secretion mechanism they used.
So yeah, so it has to ignore all that, but people are quite happy ignoring that in some of these analytical fields. And so what you end up with here is that you have these two analyses. On the one side, they say that demosponges and hexacnelids must have secreted the spicules independently because it's a different secretion mechanism. And on the other, you allow that to be ignored. If you allow it to be ignored, and you look at this version part on the right, then you see that silica is found as a shared ancestral sort of condition, feature, going all the way back past the base of the crown group of Cilicia.
Now, you have to be careful with this result because of all the problems with the analysis we've just listed, but they're getting the result that your spicules are homologous in demosponges and hexacnelids out of their own analysis.
Except that they're not preferring that method because they seem to think that that this argument that they are independent pathways and therefore independent spicules is more useful or more realistic, and so they are assigning that in the other half of the analysis, which leads to, amazingly, spicules and hexacnelids and demosponges are independent.
Gosh, who would have thought?
Which they then conclude, as it is, which they then conclude is consistent with the work of Shimizu et al. Showing that the demosponges and hexacnelids mineralize their skeletons individually.
Truly, we live in a time of wonders.
It's peculiar. Okay, we could carry on for quite a long time, but I think it's probably worth trying to draw this all together because there's a lot of confusion here, isn't there? But I think we can probably summarise this as there being three main problems, or classes of problem at least. One is that the database of the fossils, the data set that they're using, is extremely minimalist. It appears to be badly coded. There's even missed things out from which are critical aspects of the sponge biology biomineralization database. And it doesn't include anything like enough species to show that known patterns of continuous character variation that are well known among fossils in paleontology. So you end up with isolated things which give you a completely spurious tree, which means that you end up with things like Vauxia being a stem silicium, which is complete bunkum. So the arrangement of the branches is probably the most critical element in getting the right answer. And if you don't include enough to give you that basic arrangement, getting it right, you can't rely on anything else. So that's point one.
Yep. Well, point two is some of the ancestral state modelling that you then applied to this tree, and they're getting a chance of crown group racelids having been aspiculate, which is clearly nonsense. So I don't know how nonsense all the rest of the results are. Because the ones that we can be reasonably confident about are at least partly nonsense.
I think what it's showing is that there is this tendency to look at the deeper branches and smear that up the tree, which, if you are imposing at some point an aspiculate ancestor, that's going to bleed up the branches and impose aspiculate ancestor or descendants, ancestors of other groups higher up the tree, when there is no actual justification for that if the fossil record doesn't show it. So I think it is a fundamental problem with the type of analysis that was done here as well. And then you have these two different trees where you either allow the spicules to be homologous across demosponges or hexaconellids, or you don't, depending on how you interpret the results of the enzymes. And it's not even clear that those enzymes actually are responsible for the silicification. There's another school of thought that is actins, an entirely different class of proteins that do it. But anyway, so it's a very dubious thing to base the whole of your conclusions on. And unfortunately, that seems to be what they've done. So you've taken an assumption that these things must be independent, and you've got out the result that therefore these things are independent.
Yeah.
And therefore, since they're independent, you can reconcile the molecular clocks with the fossil record, because now you don't need to have a fossil record because there's no skeleton in the Precambrian.
I mean, it is possible that we're wrong, that the common ancestor sponges didn't have spicules, that demosponges and hexaconellids did mineralise independently. If someone comes up with some actual evidence for that, we're fine with that. We want to have a debate in the literature, but the debate has got to include all the evidence, and people are just ignoring half of the fossils.
Yeah, more than half.
Yes.
In this case, it was basically paying lip service to including the fossil record because there was virtually nothing included. The stuff that was included was put together for a different purpose, and it just wasn't a database that was fit for purpose for this question that they're trying to answer. And as a result, you get nonsense trees and nonsense sort of character state assignments. And okay, so overall verdict. Does this paper actually live up to its promise of reconciling the molecular clocks with the fossil record by demonstrating that spicules are independently derived in each class of sponges?
No.
Do I get a vote too?
If you like.
No.
I mean, we'd love it if someone would do this type of analysis, including all the fossil information. That would be brilliant.
And we understand that might be in the pipeline somewhere.
That would be really nice.
Yeah, so hopefully in a few years we'll actually have a decent piece of work which does address this properly. But at the moment, this paper is seriously premature in terms of drawing any conclusions of this type. And I strongly suspect that if you include the wide range of sponges that we know about and show continuity through these fossil groups that there are no modern representatives of, and show all these unique character combinations, then you end up with a completely different answer.
You mean that actually looking at the actual ancestors of the actual living things today might give you some useful information?
It might do, yeah. Allegedly.
What a startling idea.
Yeah, yeah. Yeah, so anyway, it's always nice to have a good rant every now and then, isn't it?
It is, isn't it?
So if any of the authors come across this, then they won't be surprised because they know what we think about this this analysis. And yeah, the paper's out there. It's been widely publicised. We've had quite a few people contacting us and saying that, or asking our opinion at least, on whether this really is the answer, that the problem is solved. But yeah, hopefully we've got across at least a small part, well, quite a large part really, of the problems that there are with this analysis and why we don't think it makes sense at all, and certainly can't be used as proof that this problem has been solved.
But that might be enough ranting for one day.
Yeah, I think so.
So there are rumours there's going to be more graptolites soon.
Allegedly. Supposedly. Okay, more graptolites on the agenda.
There might be a bit of a grapplite rant as well.
Yeah, but quite a fun one.
Oh yes.
Yeah, all right then. Right, we'll leave it there. Thank you for listening, and I hope that wasn't too distinct from our normal sort of level of ranting. So, yeah, see you again soon.